Good day to everyone. On behalf of Nyberman Bioinformatics, I'm Bhavani here, and I'm excited to announce the start of our 60-hour virtual internship on AI-driven omics data analysis, scheduled from 22nd December 2025 to 24th January 2026. Today, we're launching the phase 1 of the program, RNA-seq for beginners. This program aims to offer a practical introduction to genomics by combining RNA-seq analysis with essential machine learning techniques. We are eager to welcome participants from Chaitanya Bharati Institute of Technology, all sharing the passion for exploring the possibilities of bioinformatics. The objective of this phase 1 30-hour internship program is to provide hands-on training in the complete RNA-seq analysis workflow from raw FASTQ files to differential gene expression analysis. Participants will gain practical experience with key bioinformatics tools, set up and use Linux and R environment, and develop skills in data visualization and biological interpretation. The program also includes a group-based mini project to apply RNA-seq workflow in a collaborative research setting. To begin with, I would like to introduce about Nyberman Bioinformatics Europe. Nyberman Bioinformatics was established in 2019, has rapidly grown in a globally recognized bioinformatics service firm. Our mission is to unite academia, industry, student, and researcher worldwide in bioinformatics and biology. In 2021, we launched Nyberman Bioinformatics Europe, enhancing our reach and impact. We have successfully conducted over 300 webinars, each attracting over 500 participants and offering valuable workshop and skill development programs. Our commitment remains steadfast in fostering collaboration and providing opportunities for project outsourcing, consultation, freelancing, and tailored training programs. In a nutshell, we are a platform dedicated to advancing bioinformatics research and education. For more information, please visit our website www.nyberman.com. For the better quality of our program, we have emailed the internship guide to all the participants. This guide includes essential information on 3 communication platforms which we will use throughout the program. Number 1 is the Telegram discussion forum used for addressing technical questions and discussion. Number 2 is Google Classroom, where recordings and study materials of the session will be uploaded. Please join the classroom using your Gmail account, as other email providers are not supported by the platform. We also have Google Drive link attached to access input files, study materials, and datasets. We also request you to subscribe our YouTube channel to view the recordings of the session. We request you all to join all the platforms, as we are not responsible if you didn't join and miss any important update. Additionally, the guide contains Google Meet link required to attend the live session, and this link remains the same for all the sessions. The detailed program schedule can be found on page 3 of the guide. Please ensure you review this schedule and stay updated with the planned activities. The internship program will culminate in 2 assessment phases, and the details are listed in page 6 of the guide. I would like to formally say the same now. We have a collaborative group project and an individual evaluation. First is team assessment based on project presentation. A representative from each group will be allowed to present your team's work. Please refer to page 5 of the guide for group members' detail. Each presentation will last for 15 minutes. We request you to focus on your project progress as finalized outcome alone is not required. We recognize the best group and all the members of the group are eligible for 10% discount for all Neighborman paid events of 2026. We also recognize best performer and he or she will be awarded with the best performer award and provide 10% discount for all Neighborman paid events of 2026. Followed by team assessment, we have individual assessment, which will be conducted as an on-campus e-quiz. The schedule for the assessment will be announced at a later date. The overall structure of the quiz is outlined below. We have e-quiz, which used to test the understanding of key concepts and tools covered in the program. It includes section linked to an internship module. Everyone who has 60% attendance and score at least 30% in the quiz will receive a completion certificate with its score printed on it. Those who do not take up the quiz and have less than 60% attendance will receive only the participation certificate without the post-evaluation score. To encourage the collaboration and hands-on learning, participants have been divided into small groups. This will allow you to work together on a real-world challenges, enhancing both your teamwork and bioinformatics skill as you explore omics analysis. Each group will undertake a mini project to apply your knowledge. Kindly refer to page 5 for group members' details. For any queries or assistance after the internship program, please contact us at info@neighborman.com. You can also reach us through the official WhatsApp group for timely support. It's my great pleasure to introduce Dr. Saurabh Gupta, our expert tutor for the phase 1 of the program. Dr. Saurabh is a distinguished bioinformatics expert with over 15 years of experience in next-generation sequencing data analysis, single-cell transcriptomics, spatial biology, algorithm development, genome assembly, and plant stress biology. He specializes in developing analytical workflow for advanced genomics research and is currently focused on identifying biomarkers for live cancer using cutting-edge single-cell and spatial transcriptomics technique. Affiliated with Curtin University as a research fellow in the Curtin Medical School, he actively contributes to the studies on cancer microenvironment and immunotherapy responses. We extend our heartfelt thanks to Dr. Saurabh for accepting our invitation and joining us to share his expertise. Neighborman team is delighted to have you on board, and we are looking forward to learn from your valuable insight. We also sincerely thank CBIT faculty for placing their trust in us and providing the opportunity to conduct this valuable program for their students. We also extend our gratitude to all the participants for registering and engaging in this program. We are delighted to have you on board and looking forward to a meaningful and enriching learning experience together. Without further delay, I will now hand over the virtual stage to Dr. Saurabh. We're looking forward to embark on this journey together. Thank you all once again. Over to you, Doctor.
Thank you, Bhavani, for the nice introduction. Just a second. Yeah, hello everyone. Welcome to the internship program, and I think Bhavani already covered, what we are going to take you through for the next few weeks, right? So my idea is I'll take you through the first aspect of the is this internship where we would be covering a little bit about the basics of RNA-seq. So I think the way we have divided the internship is like in phase one, we'll be covering about the RNA-seq, like what it is and how do you like design the experiment, what kind of workflows are there to in a general RNA-seq experiment, and how do you analyze the data, right? And then, so that's the thing that I'll be covering for you guys in the next couple of weeks. And we would also have a mini project in the end. And yeah, also today as well, we would be making your teams also. So perhaps right in the end of this session. So I'll just share, like ask Bhavani to share me the list of all the names who have registered. And then we'll form the teams for your mini project. And then, maybe you can already, like, let's say, start maybe thinking about it, try to get your acquaintance from it. And just one question. So Bhavani, like, are all of them from the same class or different classes?
They are from same department, different semesters.
Okay, different semesters.
We have from 3rd, 5th semester students.
Okay, got it. Oh yeah, okay, you have already made the groups. Great.
We have already.
Okay, then that's good.
Divided based on their semester. All semester students are in the same group.
Okay, yep, that's good. Yeah, so maybe I will cover this one in the end. Yeah, that's fine. So this is in the end of the session. Okay, then let me jump into it straight away. Yep. Do you see my slides now? Can you reconfirm?
Yes, sir. It's visible.
Yeah, just 1 second. And also the pointer?
Yeah, perfect.
Great. Okay. All right, fine. So it's 10 a.m. in India, right? Yeah, so I'm in Australia, so it's afternoon for me already. So, okay, so good morning, everyone, once again. So today we will be learning a bit about RNA-seq. So that's one of the most important techniques used in molecular biology and biotechnology. And I reckon like you guys are in BTech Biotech second and third year, right? So I don't think like probably you would have already been covered a little bit of what the RNA-seq technique is, like, or maybe just at least heard of it, right? And our idea in this internship is to like get you close to expert level, yeah, if not expert level, like close to expert level. Okay, and in this session is specifically designed for beginners, so you don't need to have prior experience in bioinformatics or sequencing. And by the end of this talk, so you should understand like ideally what the RNA-seq is, why we use it, and how it helps us to study genes and diseases. Yeah, so just to get it, like you should be getting more and more familiar as the internship goes through. And also, so currently we have 1 hour 15 minutes left for this session, or 1 hour 45 minutes, right? So what I'll have is I have few slides, so I'll just go through them one by one, and then maybe take up the questions right in the end. Yeah, so I'll keep ample amount for questions so that if you have any sort of doubts, so we do that. And also have some slides where we would I'll get your feedback, so just to see like not anybody's not sleeping. Okay, so the agenda for this talk is what we'll cover today. First, we will see understand the different sequencing technologies in general. Yeah, like that's the seq part of RNA-seq. Yeah, so RNA sequencing. So sequencing is done by various sequencing technologies currently. So we'll understand what kind of technologies are available and how they work and stuff. Next, we'll discuss what a particular biomarker is. Yeah, so you might have heard this term somewhere, a biomarker. So we'll cover what exactly it is and why it is important to study these. Then we'll move on to the main component, which is basically the RNA-seq, right? So sequencing and biomarker is kind of a, let's say, a prerequisite before we jump into the RNA-seq. And so it will cover also what it means and how it works. And then I'll also cover a couple of examples, real life examples, and where RNA-seq is used in research and medicine. Yeah, and this is also we all have used RNA-seq in some sense. I think in the last especially 4 to 5 years. So I'll come to that when I get to that phase of the presentation. Now, before we jump into the other stuff, yeah, so these are the learning objectives. So by the end of the session, you should be able to have a basic understanding of the sequencing technologies. You should be able to define biomarkers and what are the different types. And yeah, you should be explained to anybody, yeah, that what RNA-seq and how it works is. You can even perhaps explain it to your family members, so I'll try to break it down to that level. Because yeah, so my understanding is like if you are not able to break down anything into a very like simplified manner, which you can't explain to your parents, right? Like any of your relatives, so it means you have not understood it well enough. So let's see, like if we can make some sort of a justice to that today. And then I understand how RNA-seq helps to identify biomarkers and like again how it is used in real life. And then these should be, and these concepts are very useful if you're interested in like down the line. So you're in bachelor's now, right? So let's say if you want to pursue your career into biotechnology, bioinformatics, genomics, or medical research, so then like this internship is like quite important for you. So this kind of founds a basis for it. And now just talking about the real life example. So, this is how I used to learn as well. So during my course, so sometime whenever I used to go anywhere, right? Like even in a trip or like even to sometimes do shopping, right? So I used to sometimes think in the terms of RNA-seq or like not only RNA-seq rather, like, rather whatever I'm like, being taught that time, like, try to relate. So, maybe if possible for you, so not only for this course, but in general, try to relate everything with like real life, right? And most of the things you can, so that also helps to like make improve your understanding of relative concepts, and also to for you to know that if you are really understanding it well enough, that you can relate it to this. Yeah. Okay, so now first we need to revise one important concept, that is the central dogma of life. I think this you might have covered already, but again, like, just to, so that everybody's on the same page. So central dogma explains that how genetic information flows in a particular species. Yeah, this can be humans, plants, microbes, or anything. Yeah, so where the information flows from DNA to RNA, and yeah, so DNA to RNA, and then from RNA to protein. Yeah, and so think of it this way: so DNA stores the information, RNA carries that information, and protein performs functions in a particular cell. Yeah. So depending on whatever this RNA sequence is, so whatever protein you get out of it, so a respective protein uh, performs the respective function along with it. So the RNA-seq aspect obviously focuses on the RNA stage, so which tells us that which genes are actively being expressed in a cell at a given time. Okay. So now, like all of this RNA-seq or any other sequencing technologies, they kind of fall into the umbrella of genomics, right? So what exactly genomics is? So this basically is the study of the entire genetic material of an organism. Yeah. So to give you a bit of a context, so in a human genome, we have nearly 3 billion base pairs. Yeah? So think of these 3 billion base pairs as like a long stretch of sequence. So for example, in the term genomics, right? So let's say this is a sequence. Okay, this is actually it could be a protein, so not a DNA. So how many letters are there? So there are 8, right? So 1, 2, 3, 4, 5, 6, 7, 8. So think of human genome size as 3 billion. That is, so we have a sequence of ATCG of 3 billion, yeah, 3 into 10 to the power 9. So that is the enormous size of the human genome that we have. And the genomics, what it covers is we try to study the whole 3 billion bases, yeah, at a given time. So this is what genomics encompasses. So you can imagine the scale that we deal with, especially with the sequencing technologies as well. We deal with whenever we are talking about genomics. Yeah. So, and now to give you context in plants. So in plants, you might have heard about Arabidopsis. So that is the model plant we work with for any plant-related experiments, and the genome size for that is only 135 million. So as compared to humans, right? And similarly, there are also certain plants which have even like much larger genome size. So for example, I think in the case of wheat, we have the wheat, like the wheat flour that we eat, right? So the rotis and chapatis and stuff. So the genome size of that is 16 billion base pairs. So you can imagine, like, how many ATCG bases are there and what kind of complexity is involved in this, right? So now what genomics basically answers. So the study of basically genomics answers that how these billion bases or 135 million bases are arranged in a particular cell in an organism. Now, why this is important is because this arrangement of ATGC, so in every organism, so these ATGC base pairs are arranged in a very specific order. And that order is what determines that whatever a species is doing, yeah, or let's say, or whatever the characteristics of a species are. So for example, in us in humans, we have two hands, two hair, like two ears, two legs and stuff, right? So all of this is governed by what's basically in your DNA, that is basically what's in your genome. So hence it is very important to study the complete set, like, or how basically these ATGC bases are arranged. Yeah, and so these set of so DNA, so this can be present in basically two components. So for example, the nucleus. So that is where, so when we talk about genome, so we mostly talk about the nuclear DNA. So the DNA which is present in the nucleus of a cell. But also there are other DNA available as well. So for example, we have DNA in mitochondria, and also we have DNA in ribosome as well. Yeah. So, but mostly like when we talk about only DNA, so largely it is only about the nuclear part. Yeah. And here you see, for example, in humans as well. So in humans, we have diploid set of chromosomes. So what we have is 22 pairs of autosomes, and then we have X and Y sex chromosomes, right, which determine our gender. And again, so all of this combined together, it forms 3 billion base pairs for humans. Yeah. Now, how do we study this? Yeah. So like genomics, yes, the idea is that you study the whole complete set of genetic information of an individual. But how do we study that? So we study that by using something called as sequencing technologies. Yeah. So basically what these allows is to read this order of ATCG. So as I mentioned, so we have A, D, C, and G nucleotides, right? Adenine, guanine, cytosine, and thymine. And also like instead of thymine, you have uracil and RNA. So these sequencing technologies, basically, like simply put, it just scanning through this DNA and reading it, yeah, like ATCG. It's like kind of like a reading a book. Okay. And without sequencing, it would be impossible to analyze like genomes, transcriptomes, and gene expression at a very large scale. So we used to do that like back in the days, but like before any sequencing technologies, but that used to be super slow. And the scale of it, like that time you could only study, let's say, only one gene at a time, yeah, or only very handful of genes. But now with sequencing technologies, like the scale is enormous, and you can actually study the complete set at once. So
Sorry to interrupt you.
Yeah.
Shall we get quick confirmation from participants whether they understood?
Yeah, sure. Okay.
Yeah. Dear participants,
till now.
Yeah. Dear participants, kindly give quick thumbs up if you understand the concept till now. You can also feel free to post your questions in the chat and unmute, get clarified if you have any questions.
Yeah. And again, like feel free to, write in the chat if you think like, I'm going a bit fast or slow, like I'm happy to, change my pace. Okay. And if you think like my pace is fine for now, like, yeah, okay. And again, like don't feel shy about anything. Okay. Just feel free to write, whatever, like you guys think about the speed. I'm happy to, slow down or pick up, like if you guys think. Okay. All right, fine. Then I'll get on with it. And again, feel free to use the chat messages. So I have them right in front of me so I can just see the real time and stop and have any questions, even now. Yeah. So it doesn't have I don't have to take the question right in the end. So I'm happy to take them during the presentation also. Okay. Yeah. So now a little bit about the history of sequencing. So sequencing technologies, like any technology, right? So they have evolved a lot over time. Yeah. So for example, what do we use daily? So we use technology. We are surrounded by technologies. Yeah? We carry technology in our pockets. We have our phones. So we see like, Google or Apple, they release a new version every year, and they have upgraded camera, upgraded this, upgraded that. Now, same is like true more or less for nearly all technologies. And of course, sequencing is also technologies, right? So they are not like behind. So, and they have in fact evolved even further than like, what the like all the phones are. So earlier methods, so for example, it all started in the 1980s. Okay, so one question here. So have you guys already covered what gel electrophoresis is? Can somebody just give me a thumbs up? Oh yeah, okay, great. And both second year and third year? So okay, so anybody from the second year batch? Can you guys give a thumbs up if you have already covered? Yeah, yeah. Okay, you guys. Okay, so I'm guessing that you guys have already covered, and even in the second year, that what gel electrophoresis is, right? So back in the days, so for sequencing, so in the 1980s, so we used to do sequencing just using this manual gels. Yeah, so where each row in a gel would give us one base pair, right? Now think of it, and what did, okay, anybody remember what's the genome size of humans? Anyone? Just write in the chat. I just covered in like 2 slides ago. What's the genome size of humans? Anyone? Yeah, great. Exactly. Now think of it like, so in the back in 1980s, so we used to how sequencing used to happen. So we used to follow gel-based systems, huh? So we used to have gel electrophoresis. So initially, when we started, it was just like manual gels. And now think of it like in each gel, how many different rows you can have, right? So I think like perhaps like only few, I think less than even 100, or let's say if you have a very big gel, you might go into like, few hundreds or maybe a KB or something. I think not even that. So for example, here, if you see, so this tells you that that time we could sequence 10 kilobases per day. Yeah, but this is again like, using manual gels, like, you form different gels, you run different ladders, right? And then you try to see like what the sequence is. Now try doing this for a 3 billion base pairs. So you can just imagine that how many number of gels you would need if you have to actually follow this manual gel. Yeah. So like we humans have been here forever, more or less, right? For like many thousands of years. But like we weren't able to, really understand or study humans at that scale, especially at that time. Yeah. Of course, it is a good start. Yeah. So in 1980, at least, we started doing these things, right? We like somebody at least thought of like how we could sequence, right? So that was again a great start. But again, like if you compare from then to now, it was like close to impossible to, like do this stuff. Yeah. So then over time, as like sequencing technologies evolved, so we like by 1980s and stuff, so at least we went into like automated gels. Yeah, so there were ways to do like gel electrophoresis a bit in an automated fashion. So that was great. And because it was automation, so you can imagine like, the manual intervention was a bit less. So this also it resulted in the increase of throughput. So throughput basically means like how much you can sequence at a given time or a day, yeah, or from a single run. So you could sequence like much more. And then came something called as capillary sequencing. So that is basically what we call Sanger sequencing, right? So this started in 2000, and this even further. This is again like another sequencing technologies which is now also used a bit routinely as well. So as a validation purposes. And so this even further increase. So if you see that the y-axis, it's actually logarithmic scale. It's not a linear scale. Yeah. So from 10 to 100, then 1000. So this is actually you are going on a logarithmic scale. The throughput of the sequencing technologies that just kept increasing and increasing. Yeah. So capillary sequencing that increased like much further, and then the real boom happened actually after 2005 when the next generation sequencing technologies came up. So the first one came was from Illumina. So I'll be talking a little bit about it in the next slides as well, right? So basically, where I'm trying to get to is, so earlier methods, they were slow, expensive, yeah, and low throughput. And over the years, improvements have been made for faster sequencing, that is already, and this also results in lower costs and also higher accuracy. Yeah, so this has also been increased because in the gels, like sometimes it's not so easy to do it at the scale, but whereas in the NGS sequences, the accuracy these days is quite high, right? And this progress has also made techniques like RNA-seq possible and accessible to researchers worldwide. Yeah. Now to give you an idea, like one sample of RNA-seq or sequencing, like back in the days, would cost okay, actually I have that slide after 1 or 2 slides. Maybe I'll come to that then. Okay, so now with that history of sequencing, so just again, like, how we address, so what is sequencing? So that basically just determining the order of A, T, G, C, and U in DNA and RNA, right? And without sequencing, what we see, we only see the phenotype, yeah, or symptoms of protein or traits, right? So for example, we as any humans as well, so somebody has black eyes, blue eyes, right? So the eye color is different. Somebody has brown, dark brown, or different shades of brown. So all of these are governed basically by your genes, yeah. And by genes also basically is like the order of the nucleotides in your genome. So that's basically a phenotype. And without sequencing, so we would only know this. And once if you sequence it, and then we would also know basically is the genetic blueprint. That is, then we can associate even the phenotypes with the genotype. That is like which of these sequences, which order of the nucleotides, and which place in the genome governs this particular phenotype, is responsible for this phenotype, right? And then in terms of the nucleotides, I think you already know. So we have adenine, guanine, thymine, cytosine, and uracil for RNA, right? And here it's just like very brief overview. So this is from liver. So where you can extract basically a tissue from whatever source of your tissue you have. So in this case, you have liver, you extract the DNA, and then you prepare library. So in sequencing technologies, you have to first do some sort of a pre-processing of your samples to make it compatible to the sequencer. So sequencer are the machines which actually sequence the DNA. But to feed your DNA into the machine, you need to pre-prepare them in a certain way, which is called libraries. So once you extract a DNA, you have to prepare a respective libraries into it, and then depending on sequencing technologies, you can make PCR copies using PCR, and then feed it to the sequencer. And then once you get the data, like basically the order of sequences, and then you do your analysis. Yeah? So this, in a nutshell, is what sequencing is. And so basically, sequencing is like reading biological instructions written inside the cells. Okay, and these instructions are what determine our, let's say, phenotypes, for example. Okay, everything clear till now? Great. Okay, now what do you think is the cost of sequencing this? Yeah, so I told you a bit about the size of the genome, and I don't know if you guys would know about the sequence cost of gel electrophoresis, but again, any guesses? Yeah. So what do you think would be the cost of sequencing the entire human genome? And just to tell you, give you a bit of a background. So the first human genome which was sequenced, so that was sequenced by spending millions of dollars, and that took several years. Any wild guess? What's the cost these days? Any number? Just give me any number. What you could think. It may be this. Just a wild guess. Anyone? Just type it in the chat. Okay, no one. Okay. A few lakhs. Okay. Getting there. Okay, definitely not in millions of dollars. So yeah, that helps. Okay. Anybody else?
Dear participants, feel free to post your answers in the chat if you cannot unmute.
Yeah.
Please make the session interactive, guys. This is not your academic class. We are not our faculties.
Yeah. No, okay. I just one more answer. Anybody? Before then I'll move on. Just give me anybody one answer. 1 billion. Okay, Bhavan, tell me one thing. Have this tens of thousands? Okay, no, wait. Pratima, I'll come to you. $1 billion? No, it used to be $1 million before. That was the first one. And in the sequencing technologies, I mentioned that the sequencing cost has come down, right? So would it be more or more in the tens of thousands that Pratima mentioned now? Okay, fine. Then in the interest of time, to move on. So Pratima, let's say you are the closest among the three of you. So at the moment, yeah, so this is first of all a graph, yeah, so to show you over time, and this is also till 2022 at the moment, that how the cost of human genome has decreased over the years. Yeah. So for example, like the first human genome, which was in 2001, if you see like how many dollars, 100 million US dollars. And that is like, okay, I'll have to calculate how many crores that is. I think close to 900 crores or something, if my calculation sounds right. Yeah, so that's about like, how much it costed to sequence the first genome. And now, so first of all, before I go to this graph, so in in computing, yeah, so there's something called the Moore's law. Has anybody heard of it? Moore's law? No? Anyone? Yeah, okay. So in computing as well, right? So again, that's technology also. So in computers, so what we have is, so there was a time like by Moore's law, it used to tell us that every year, right, the number of transistors that you can have on a given area of a chip, it doubles. Yeah. So and the number of so basically a transistor is what actually is responsible for your computing of any kind, right? And if you're able to multiply or double the number of transistors, so that also tells you that with using the same size of chip, you can actually compute more. So if you see in the starting days as well, so from 2001 to 2007, so more or less the sequencing was also following kind of this trend that over Moore's law. So the idea was that every year the number of transistors that can be placed on a particular chip doubles. So this would also bring the costs down. And this was a projection here as well that, with the sequencing also, that the sequencing would follow the Moore's law, and these numbers would follow the similar line and keep decreasing at a logarithmic, like halving every year. But it was fine till the first time, where we had the sequence gel-based methods or capillary-based methods. Now, in 2005 is when the first next generation sequencer came out, so which was basically from Illumina, but it wasn't like still in the because of the first one, and it was still relatively costly. So it took some time for the cost to come down, but once they did, so and they really came down like quite a bit, because this is also because over time there was a few other players as well, sequencing companies, which started coming up with their own sequencers, which resulted in a kind of a price war as well, if you may, for sequencing technologies. And then the cost dropped down quite a bit. And then because it was dropped down quite a bit, and you can imagine that it is getting into let's say $10,000 in 2021. So now this is really getting into the bracket where it is affordable for more or less anybody to sequence the genomes. Yeah. And now because they were able to do that, so hence the number of amount of data being generated also increased rapidly. But again, you are sequencing the genome. Yeah. Again, like in humans, you have 3 billion base pairs that you have to sequence, right? And we also sequence in multiple times. So it's not that we sequence the whole genome in one copy. So we copy the genome and we do some PCR duplicates, and also we sequence the genome multiple times. So you can imagine the amount of data which is generated. So 3 billion bases is roughly let's say an uncompressed format is 3 GB of data, right? And these days we have sometimes okay, back in the days we used to have pen drives of 4 GB or 8 GB, right? So these days in terms of NGS, we deal with data amount of few terabytes. Now where I'm getting to it is, now the computing is following Moore's law that the number of transistors is halving every year, but we are generating even more sequencing data than what the computers can handle. Yeah. So now we are coming up with this was back in the days in 2010 and stuff. So but over the years now we have come up with some smarter ways of even analyzing the data, because we couldn't keep the computing powers, they couldn't keep up over time, right? So this just tells you the massive reduction and first of all the cost, and this also imposing a challenge from a computing perspective, that because the cost came down, so everybody is able to sequence, and or we are able to sequence more. But that also results in more let's say computing power or more smarter ways of computing needed, right? And this massive reduction is also the reason in cost is why RNA-seq is now widely used in cancer research, drug discovery, and personalized medicine, right? And this is and we'll be covering some of the examples for this as well. Now, just to give you a little bit more about the brief history of sequencing. So as I mentioned, the first generation of sequencing was called the Sanger sequencing, so which was developed by like Frederick Sanger in like now 1980, so 77, right? So which uses the chain termination reaction where we add fluorescently labeled nucleotides. And every time a new nucleotide is added, so it releases a certain fluorescent label. And this label is captured, and based on which color of fluorophore is released, we say that if it is A, T, C, or G. And that time we could sequence like up to 800 bases or 1000 bases. And the strength was it was highly accurate. And it used to be, and it still is the gold standard for small sequences. And the weakness, of course, like, as you can imagine, these were like manual gels. So it was extremely low throughput. So you could only sequence, let's say, one gene at a time. And this was also used for early sequencing of viral genomes and validating mutations. And still we do Sanger sequencing to validate the mutation. Yeah. So here is just a schematic example of how it looks like. That way, you run these on. So every time a new nucleotide is added, so for A, T, C, G, we have different fluorophores, which are released whenever a nucleotide is added into it. And then we run all of these on a gel, in a, let's say, capillary gel electrophoresis, and then you put a laser beam, and then you get this sort of a chromatogram. And then by reading this chromatogram, you can actually determine the sequence of your sequence. Yeah. So this is how it used to be back in the days. But yeah. Okay, everything clear till now? Great. Yeah. Now, but then over time, right? So we came up. So in 2005, we moved on to something called as next generation sequencing. Yeah? So here I'm showing you some of the examples of the sequences which are there today. So these were not the first sequences from the different companies. So these are some of the major platforms. So this was the one NovaSeq 6000 from Illumina. Then this is from another provider from China, that's MGI. And then this is from Oxford Nanopore, and this is from PacBio sequencing. So what all of these sequencing sequences, they come under the umbrella of next generation sequencing, which actually allows massively parallel sequencing of DNA or RNA. And yeah, so next I'll just go one by one. So a typical sequencing workflow, right? So so this slide shows you the typical workflow for that. So first, the RNA. So in terms of the RNA sequencing, right? So RNA is converted into cDNA, and then you add your, like adapters on both the ends, and then you linearize or enrich using PCR, and then you do sequencing. So this is just a very crude, rough overview of how the sequencing looks like. So this is true for all of these, different sequencing technologies. Now, it might look a bit complex, but it's not really that complex. So the idea is very simple. Yeah. So in terms of RNA, you convert that into cDNA, and then you do a bunch of stuff to to load this converted cDNA into your sequencer, right? So for that, so remember I mentioned earlier about preparing a library for sequencers, right? So these steps basically are those to before loading your cDNA onto your sequencer for sequencing. So you have to do this steps as a library preparation. So every sequencing company has their own steps to follow. But again, these are all standard protocols which are given by the company, or you don't even have to do that. So these days, you mostly you would get your sequencing done from a company. So all they would need is just the RNA in, let's say, cDNA format, for example, because that's more stable than RNA alone. And you just give the company the cDNA, and then they will take care of everything and give you the data. Okay. Now, so first, now I'll talk to you a little bit about Illumina. So that is basically the major sequencing company provider worldwide. Right. So this company, Illumina, so the sequencing that they do, so this follows a principle called as sequencing by synthesis. Okay. So this is similar to how we do for gel electrophoresis, where so each time a base is added, a fluorescent signal is detected, and this color tells us which base has been incorporated. Okay, and by repeating this process, the entire sequence of a fragment is read. And yeah, so then this is like quite reliable. So now this company as well, so that's basically what sequencing by synthesis is, that you keep synthesizing the second fragment one by one, and every time a new base is added, you detect a particular fluorescent signal, and then which determines what is the sequence, what is the base which is being read. Now, the first platform that came out from Illumina was in 2007. That was called the Genome Analyzer. So at that time, it could sequence only 30 bases of a given read. So the way we do in all these NGS sequencing technologies, except for two long read ones, so which I'll talk about later, is that you take the whole full DNA of your, let's say, organism or species, you break that into small pieces, small fragments, yeah, generally in the order of like a few hundreds, yeah, 100 to 200 fragment size. Again, depending on sequencing technologies, it varies. And then you read each fragment parallelly. Okay, and this is what it tells you. So each fragment is read. The 30 bases of each fragment are read, and later on, like within the genome analyzer itself, they increase this to 100 bases per read. And about the massively parallel aspect, so that time you could sequence up to 6 million fragments per lane. So how you sequence the your DNA or RNA on the sequencer? So this is for Illumina. This is how a standard, how this is a flow cell from Illumina. This is the size of a standard glass slide that you use in your microscopy as well. Yeah, so that's basically the size of it. But the way it is defined is, so it is divided into multiple lanes. Yeah, and you load your sample or your library into these individual lanes. Yeah, and here for a scale, you can imagine that you could sequence 6 million fragments per lane in parallel. Yeah, so these are examples of some of the sequences that are there. And then over the time, like they even also improve this further with, let's say, the second platform was HiSeq series in 2010, and these days now they have currently a NextSeq series, so which basically had a slight change in the sequencing chemistry of how they sequence it, just to again address some of the accuracy concerns. And here you could actually sequence 4 million base pairs in a 30-hour run. Yeah, so the sequencer would run for 30 hours, and then you could actually sequence 400 million base pairs. And this even improved further by NovaSeq and HiSeq X10. So, and in today's era, you can actually sequence 16 terabytes of bases in a single run. And this had also helped to bring down the human sequencing cost to $1,000 or even less than $1,000. Yeah. So now you can imagine the amount of the scale at which we are talking about in terms of sequencing. Like now you really see the massively parallel aspect of sequences. Yeah. Now here I have a couple of slides now to give you a little bit of how exactly it works in Illumina. Yeah. Again, I'll try to simplify it as much as possible, but again, you don't have to stress too much on the technical details, but because again, all of these are standard protocols, and you don't really have to do this. Like the sequencing technology companies, they take care of this. But again, you should still get a feel of how these things are done. Now, as I mentioned, so on a flow cell like this, so you have 8 different lanes. And on each lane, what you see here, so think of this component here, right? So forget about this part yet. Yeah, so you have this some oligonucleotides which are attached to this glass slide. Yeah, so these are physically attached to the glass slide. And what these fragments contain, they are the complementary sequences of the adapters which we added to our DNA sequence. So earlier we had the cDNA sequence, right? So the double-stranded. So for RNA, you would have cDNA, and for DNA, you just have the standard DNA. So where we added these sequencing adapters, which we call them, on both sides, and on the glass slides, you have the complementary sequences for this. So when we load our sample, the library which contains our fragments and which has the both complementary adapter sequencing on both sides onto the flow cell, then because we have the complementary sequence already, So then each of these fragments which we load, so then they tend to bind to some of these fragments. Yeah. And the way we make this flow, like how we add the sample, so this is also done in a very methodological fashion, that we don't just load all everything we have. So there are also, like, some recommendations that how much concentration of your samples you can load. And the idea is, so we try to randomly place the fragments all over the glass slide, and allowing for like some space between them. Again, this is something like, everything is at random, and mostly the recommendation from this company, from the sequencing provider, so they take care of it. And after this step, once we load, so the what you see here, like, these is one fragment, and which is attached to its complementary sequence on the glass slide. Yeah. And then these so what happens after this is, so once these single-celled fragments are added, so this basically part is now we are getting into something called as cluster generation in Illumina, right? So where each DNF fragment, right? Now imagine this, that so how do we detect the A, T, C, or G? So that we detected that when a fluorophore signal is released whenever a nucleotide is added, right? Now, think of it like the DNF fragment is super, super, super small. Yeah, it's actually at a scale of nanometer, right? Now, detecting a fluorescent signal from that is super difficult, right? Even for, like, all these sequences as well. So first, what we try to do is we try to amplify this signal. So what we do is, so this step is something called as cluster generation in Illumina, where we try to make multiple copies of the same fragment where it has bind. Yeah. So here the way it works is, so we do something called as a bridge amplification. So where, because we have the complementary sequences attached on the glass slide everywhere, so this one complementary sequence, it is attached here. So then the fragment bends, and then it follows, goes to the other side. And then we have let's say and we add other enzymes to incorporate the nucleotides on the other strand. Yeah. And now once that is done, so now we have two copies of the same fragment, right? And we keep doing this process again and again few times until we get some sort of a clusters for the same fragment throughout the glass slide. Okay. And so now what this basically gives, so all of these fragments are identical copies of each other. And why we have done this is only for the purpose that when we add the fluorophore, right, when we do the actual sequencing later, when we read the sequence, that the signal which comes out, which would be we would be able to detect it. Now, once we do this, now we add our labeled nucleotides. Yeah, so where we have the different fluorophore attached to A, T, C, and G, and now they are added on the whole glass slide. Now, for each cluster that we form, so because they are the whole cluster is a unique read, like this, and this is a unique fragment. So every time a nucleotide is added, so every cluster will give a different color, different fluorophore, whenever a new nucleotide is added, right? And then this is what is captured by a laser, right? Onto that, so the laser captures like the whole glass slide and sees that, okay, where we are getting what kind of fluorophore. And then let's say, and this process is repeated again and again. So let's say if we are sequencing 100 bases, so then this process is repeated, like, I think close to 100 times. And then you see that every time a new nucleotide is added, that what color is coming from which location on the glass slide. Okay. And this is how, like, this ATC different color combinations here is just for illustration. And this is how an actual picture looks like from Illumina, like where, for each glass slide. So every time a ATCG is added, so you get this sort of a picture. And then from this picture, the sequencer determines that from this which particular location, so it knows. So that, let's say, take this point for example. So at this point, we would be getting different ATCG signals for the 100 cycles which we would be getting. Yeah. And now, because we know that exactly at this particular point is one fragment, so every time a different fluorophore comes in, so we just keep adding it. Yeah. So for example, here, once we get a citation for C, G, C, T, G, A, etc. Right. So here, then you get the sequence of the read from this fragment is your G, C, T, G, A. Similarly, if you let's say try to make a sequence of this particular fragment from this location, so here you get A, G, C, C, G. Okay. Now, before I move to the next step, so any questions till now? So this is just like, an example of how Illumina's sequencing technology actually works. Yeah. So any questions so far? I know like this might be a bit tricky to understand, but like, yeah, I'm happy to take questions here, like, and spend a few minutes if something is not clear. Like, feel free to unmute yourself or like, put it in the chat, or if everything is clear, like, feel free to give a thumbs up as well. Anyone?
Dear participants, do you want us to repeat this again? Hope all attending this session, right? Seems everyone is silent. Physically
I
present.
hope I haven't made you guys sleep, or
Maybe physically present
okay.
at the back end.
That's nice. It's okay. I was also like I also did my BTech some time ago. I'd never used to like ask so many questions, but again, like feel free to like this is also for your understanding. Yeah.
Unmute and answer. You guys can give thumbs up if the concept is clear. Still no thumbs up also.
Okay, so basically each fragment is going to give different colors due to additional. So each different color for each cycle. So for each fragment, so here, for example, so take this picture as an illustration, yeah. So here you see this black and white, but actually it's not black and white, so it can actually determine like what is the different fluorophore coming out. So each place in the glass slide, right, is the fragment is determined by the location in the glass slide. Yeah. So for example, in this example, what you see is 4 locations. So you are actually looking at 4 different fragments. So every time we add the nucleotides at every cycle, right? So you will get a different signal from each location because each fragment is different. Okay. Unless they are, let's say, identical copies, like which is highly unlikely, right? So those would have the same order of fluorophores. So the difference is not the different colors, but it's basically the different order of the colors over the 100 cycles. Okay, so for each fragment, because it will have a different sequence, so every time we add a color, so for example, if these 4 fragments were exactly same, right? So then you would get the exact same pattern every time. So you would get a yellow hair, green hair, blue hair, red hair, in all the 100 cycles, right? And this is how you sequence. So okay, so maybe Hadi, tell me one thing. So take this particular region as an example, and can you type in the chat what would be the sequence of the read coming from this region? Feel free to unmute and like speak as well from this. So for example, here the sequence is G, C, T, G, A, right? So what would be the sequence from this particular location, the read coming from this fragment? So green is T. Yeah, you can take examples from here. So green is T, red is A, blue is C, and yellow is G.
So for A, it would be
T. Yeah, perfect.
For T,
Yeah, now you got it. Yeah, so Manaswini has typed it. Hadi, is it clear to you?
Yes, sir. So for
You
T,
say you couldn't hear you.
A, right?
Saurabh, Bharti is trying to speak something, but I think her
Yeah,
voice.
yeah, volume is low. Yeah.
Yeah. Hadi, could you please be a little bit louder?
Is it clear now?
Yeah, better.
Yeah.
So So for the sequence, actually, I'm not able to see it clearly, like those 5 slides, like
Which one? So this slide you were able to see, right?
yeah, like they're like small T after T.
Okay, so see, the green is T,
Okay.
red is A, yellow is G, and then you have blue is C. Okay? Now, in this particular case, so here for this fragment, you would have what? T, then you would have A, then A, then G, and then a T. Okay? But you understood like how the fragments are, like how different fragments would give different order of colors, right?
Yes, yes. Yeah,
Yeah, so that's the most important part. Yeah.
yeah.
Okay, great. Okay, thank you guys. Fine. Then I will move further. Yes. Next slide. Yeah, so the next sequencing technology that I'll be covering is PacBio sequencing. So this is again another NGS sequencing technology, which is also called as the third generation sequencing technology, right? So this is works like a bit differently than, like quite differently than Illumina, right? So what this does is, they do something called as SMRT sequencing, which is actually single molecule and real time sequencing, okay? And the best part of this is they can actually sequence very long reads. So for example, they can sequence from 10 to 25 KB bases in a single read. Yeah, and this is like quite crucial, especially like if you have to do genome assembly related projects, or if you have to detect isoforms, yeah, in RNA-seq. So that is like quite crucial, and it also allows you to do precise identification of isoforms. Yeah. So what this is also the current state of the art, but the only issue at the moment for this technology is it is super expensive. Yeah. So so I did my PhD in Germany, and that time also we did a sequencing of one of the plant, which had a genome size of 1.4 GB. Yeah. So 1.4 billion base pairs. And I think that time we spent how much money? Any guesses how much money we would have spent to sequence PacBio sequencing for a plant genome of size, 1.4 GB? Just one sample. Any wild guess? Thousands, lakhs, crores, anyone? Okay, anyone, just give me any number. Even 100 rupees. No? Okay, fine. No, fine. I'll answer. Yeah, because to move on further as well. So that time we spent close to 80,000 euros to do sequencing using this technology to assemble our genome. So, and 80,000 euros in today's world, today's pricing is, what is it? Yeah, 84 lakh. So 84 lakh rupees just to do sequencing with this technology and and another one. But we spent the money because it is like quite important, and we were able to assemble a very high quality genome. But yeah, so that's for another time, because the ideas for RNA-seq today. But okay, so but this is also like one of the latest instruments, which is called Review. Now, how this sequencing technology actually works? So this technology, it doesn't need very small fragments. So in Illumina, we try to break down the fragments into like only 100 to 200 bases, or sometimes like up to 250 bases. But whereas in PacBio, because it can sequence 10 to 25 KB bases at a stretch, so you can actually break down, like you can have very larger fragments here, like when it comes to DNA. And whereas in RNA, because like 10 to 25 KB is sometimes the length of the whole RNA, right, of an mRNA. So hence you don't need to do any sort of a fragmentation there. But in terms of DNA, you would need to fragment it. And then what you do is, so like in the case of Illumina, you do some sort of end repair, and then you add ligation adapters. Now, here, something is different here than Illumina. Like in Illumina, we had open-end adapters, yeah, so on both sides, which allowed for these fragments to bind to the flow cell, right? Whereas in the case of PacBio, we have circular fragments which we add on the both sides. Okay, And in the next slide also I'll show you like how this works. But the way it works is, so you add the sequencing adapters here, and then you anneal your primers, yeah, which allows for polymerase, so which is used for sequencing in this particular sequencing technology to keep sequencing. And now because of these circular adapters or closed adapters, this actually made this particular fragment into kind of a ball, into a circularized DNA, right? And then in this particular sequencing technology, the sequencing is caused by polymerase. So the primers that we added, so then we run polymerase to add to this. And then because it's a circle, so it keeps going on and on and on in circle up to whichever time we need the sequencing to go on for. So in this particular sequencing technology, you run the sequencer for a certain set of duration. So it can be like few hours, like 2 hours, 3 hours, or sometimes like for 30 to 40 hours, right? Depending upon how much more like you want to get accurate. Now, the good thing that why we use polymerase are like there is a pro and con to that. Pro is, so polymerase, because they have like high fidelity, so they can like keep doing sequencing for a while, because sequencing the polymerases are quite efficient. However, they often tend to have many errors. That polymerase is known to introduce several errors when it is trying to sequence, right? Now, what this technology does is, because it allows you to sequence the same stretch again and again and again. So for the same fragment, you get multiple copies, but you do have errors as well, which is produced by polymerase. But what is the beauty of these errors? So these are all random errors. So it does not produce errors at the same base pair in the fragment. So every time it goes, so it would generate an error randomly. So it can be at any place. Now, but because we are sequencing it multiple times, right? So it can be like 10 times, 20 times, 30 times. So then you combine all of these data together and you find a consensus sequence. Now, with this consensus sequence, so what additional advantage you get is all the random errors are gone, right? Because in the consensus, so let's say in the first position, in this example, there is no error. Whereas in the second case, so you have 2 errors, and we have read how many times? 1, 2, 3, 4, 5, 6, 7, 8, 9 times, right? And out of the 9 times, 2 times we have errors, and 7 times we have not. And plus, it is the same sequence every time, supposedly. So 7 times we have one base, so that tells you that yes, this is the base over here, and then that error is gone. Okay, and that polymerase helped us to like keep sequencing in multiple times, and it can even sequence like very long fragments. So that is why polymerase helps. Okay, and then once we do this, so this consensus sequence that we get is what we call as high-fi reads, so which are something called as high fidelity reads, which the PacBio calls it, right? And another advantage we also get in this sequencing technology is, so which I'll show you in the next slide, is within the same fragment of read that we have read, it also gives you a signal if a particular nucleotide is methylated or not. Okay, so this I'll cover up in the next slide. So this technology actually allows 2 things. One, like 2 advantages over Illumina. So one, it can sequence very long fragments, right? Second, within the same fragment, using the same data, you also can determine the methylation status. Okay, so then basically you don't need to do epigenome sequencing if you're interested in it. Like using the same, using the same data, you can actually know this epigenome as well, so which is an added advantage for your purpose. Okay, and now how it does the sequencing? So here we have something called as wells. So here also you have some sort of a chip, like you have in the case of Illumina, you have a flow cell, right? So here also you have a flow cell, which is in the form of a square chip, and on that chip you have these small wells, and in each well you have a polymerase which is embedded at the bottom of the chip, right? And we load our samples in such a way that each well gets just one fragment. Okay, And that is how, like, we load the concentration so that, randomly whenever the DNA goes in, so only one fragment goes into the wells. And again, like similar, the concept is similar, like where you have, let's say, the different fluorophores for the different nucleotides, and you keep adding that. Now, because the polymerase is embedded below, and so and because we have well, so the good thing is we don't need to have a really amplify our data like we used to have in Illumina. So in Illumina, we did something called as cluster generation, right, to amplify the signals. However, here, because we have everything in a well, so whenever a new base is added, so we can, very precisely detect it. Yeah. So because of the, nature of how the technology is designed, and hence, you sequence everything is in real time, and that too, like this exactly the same molecule. So there isn't much of, let's say, library preparation involved in this. So the main only library preparation is adding the adapters on both sides to circularize the DNA, right? And then the same thing happens like that I showed you previously. So then the polymerase is here, and the whole fragment just keeps going on and on and on through the polymerase. And every time a new base is added, so you get a ATCG. Yeah. And then you get a signal and you sequence the genome. Yeah. So that step remains same like in Illumina, like how you get the sequencing, right? And now I'll cover just 2-3 more slides, and then I'll again stop for more questions, right? So now another sequencing company, which is again like very popular today, is Oxford Nanopore, which was founded in 2005, and their first product was a minion. So you see this on the hand. So if you can imagine, so this is, let's say, more or less the size of a USB stick that we have, a pen drive that we have, okay? That is the size of a sequencer, okay? So this is like, quite impressive. So this is also another third generation sequencing technology, right? And here in this, what we do is, so it basically passes your DNA or RNA through a small nanopore, okay? And so, yeah, so first of all, like before I get into this, so current technology sequencing chemistry that they do is R10.3 nanopore, which was released in 2020, and it also has a very high accuracy. And this also allows very ultra long sequencing. So which ultra long sequencing means, so here it can do actually 100 to 200 kbs also, okay? But the only issue is like it is difficult to resolve single base resolution sometimes. Yeah, but this is also, this solution probability is also being, like, this issue is also being addressed lately. So this is how the technology looks like. Yeah. So you have a single nanopore, and you pass your DNA fragment through this nanopore. Okay. Now, what is there in the DNA? So you have adenine, A, T, C, and G, right? And the structure of this A, T, C, and G is different. Yeah. So the weight that each nucleotide carries, so it has different kilodaltons, right? Now, how the sequencing works is, so you have this nanopore, and then you pass a current through this nanopore, okay? And then you pass your let's say DNA, okay? Now every time at here, so you have some sort of a electrode over here. Every time a different nucleotide passes through this electrode, the nanopore is like, changes a bit differently. Yeah. So think of it like there is a very tight pipe, okay, and you are passing some sort of a marbles through it, okay? Some marbles are like, small, some marbles are big, right? And then when they pass through the pipe, some pass through very easily, or there would be a bit of a bulge in the pipe as well, right? So similarly, think of it as that you're passing the different nucleotides as well. So they also may be different, vary in the size or the shape, right? So here, whenever a different nucleotide would be passed, so because of the size of it, the difference in the electrical current would be different for each nucleotide, okay? And that is what is detected over here. So for example, here, so when there is a T, so you would get this much current. If you have A, then this much. If you have C, then this much, and G, and so on, and etc., etc. Okay, and then based on this change of flow of the ions from one pore to the other, right? So this from one membrane to the other membrane. So this is detected in the form of electrical currents, and then this is translated into your sequencing read. Okay, and this is what the Oxford Nanopore sequencing, like how this sequencing is, and it was great. Yeah, so how the sequencing technology came in, but also like very in the initial days, like the accuracy was not very efficient. Yeah. So for example, like it was not able to very accurately determine sometimes the current signals, and this has been addressed over the years. But again, like still this sequencing technology is like super powerful, and it was that powerful that this technology was even used in space. So the International Space Station, right? So which hovers around Earth, right? So because of the size of the this sequencer, like what is the size? Just a USB stick, right? And on the International Space Station, they keep doing different experiments, right? And let's say they capture some sort of a soil from, let's say, a moon or Mars or whatever. Yeah. In ISS, they might have samples from whichever places, they have been surveying around. So they even did like nanocode genome sequencing right on the ISS. So where they plugged in the sequencer in just their laptops, and actually they were able to get the data directly and even sequence it. Yeah. So this just shows you how far we have come up with the sequencing technologies, right? Okay, now I'll stop here for a bit and for any questions for anything till now. And again, so this is just for your understanding to see like how the sequencing technologies work, right? So you don't have to be very very let's say that you have to know all the details. Like I mean, you don't have to worry about the very technical details, because again, like there are very standard protocols to make the libraries and sequence it, right? But again, any questions? For PacBio, Nanopore, or is everything clear? Thumbs up if it's clear. Great. Okay. Just curious, like has sequencing been taught already in any of your courses? Okay. First, I'll ask the third year students. Has it been covered? Sequencing in any of your lectures? Oh, it has. Okay. And what about second year? No? Okay. Is there some thumbs down in Meet? I don't know. Yeah, maybe thumbs down if it is not covered for second year students. Oh, it is. Okay. Okay. So then, but okay, well, that's good. Yeah. So now, what are the things to consider for if you have to design any of your, sequencing experiments? So this I've kept it in general, so not only for DNA and RNA, right? So some of the things, which you can do actually with this. Oh yeah, one more thing. So all these sequencing technologies, right? So think of it this way. So what's first of all a PCR machine? So PCR machine just allows you to amplify a fragment which you are giving it, right? And that fragment can come from anywhere, as in it can come from your genome, or like cDNA, which comes from your RNA, or you, let's say, captured a particular fragment using some sort of amplicons, right? And then but the principle behind it is that PCR machine, it just makes copies of whatever you give as input, right? Now, take that as an example in the sequencer. So sequencer is also the same in the sense that whatever you give it as an input, it just basically reads it and tells you what is the order of ATGC in that. And you can do this for any purposes. For example, you can do this for whole genome sequencing, for transcriptome sequencing, target sequencing, epigenomics. Everything is done on the same sequencer. The only thing which changes is actually your library preparation protocol. Okay. And then sometimes you also have to consider what kind of sample type you have, or what is the sample quality. So some platforms have higher or lower DNA or RNA concentration, and some are better suited for handling degraded samples or lower quality samples, right? And then also what kind of analysis you need. So that determines what kind of read length or sequencing strategy you need. So for example, if you have to do genome assembly and your genome size is quite big, so then you are better put off with PacBio. But again, you have to be a bit wary about the costs there. And then of course the cost is a very big factor, like especially in the Indian context, where it can be quite expensive, especially if you have to do a whole genome experiment. And then of course, because you're dealing with terabytes of raw data, so you have to keep in mind if you what the computational architecture infrastructure that you have available. So tools available, mostly you don't have to worry too much about it, because more or less for all the tools, for all the sequencing technologies, more or less there are tools available, right? So that part is now more or less okay. But again, computation infrastructure is something still, you need to be a bit wary about. So RNA-seq and stuff standard, I think you can also do on your laptops. But again, for your other genomics MD related stuff, you would need like much higher computation resources. Now, this here, I just try to summarize all the sequencing technologies that we covered. Like we have Sanger, Illumina, PacBio, and Nanopore, right? So basically, the short read sequences like Illumina, so they are best for quantifying the RNA. So where you don't really need full length sequences unless you are interested in detecting the isoforms or splicing, because and if you are, so then you're better off doing PacBio. But again, the cost would be quite high. So that is something you have to be a bit wary about. And if you have to do like splicing or let's say some sort of a field work or some real time data, so then you would be better off in PacBio or Nanopore rather. Yeah. Okay. Now a question for you guys. So if you want to sequence the transcriptome of a bacterium that has operons and overlapping transcripts, which of these technologies you would use and why? Anyone? Just raise your hand and unmute yourself. Or just unmute and speak away. Give it a think. Anyone? Let's say money is not a problem, yeah? So you have all the money that you have. Yep, exactly. And why? Yep. Yep. Both correct answers. Okay. Okay. Now it means you guys are understanding. So that's good. Okay. So now we move on to the next part of the presentation. So which is about the biomarkers. Yeah. So here, I have a couple of slides for to give you just to cover a kind of a concept basically. Yeah. So a biomarker is basically a measurable biological indicator. Yeah, so it can be like a gene or a RNA or a protein or a metabolite that tells us about a particular disease or the normal biological processes or response to a certain treatment. Yeah. So, for example, let's say you do some blood tests, right? For let's say detecting anything, maybe say inflammation or measuring diabetes and stuff. So what you actually are doing is you're getting some sort of a biomarker checked. Okay. So then there are different kind of biomarkers available. So they have diagnostic biomarkers, prognostic, predictive, and pharmacodynamic. So prognostic basically means biomarker which can help you to determine that if somebody has a disease or not. Okay, and so based on the presence or absence of this particular, or let's say this biomarker, of it can be either, yeah, it can be like DNA, RNA, or protein, or metabolite. So this determines that if it basically allows you to screen your healthy patients from your, let's say, diseased or cancerous patients. Okay, and then we have your prognostic markers, so which actually are which estimate the risk of it. So which don't really tell us if the disease is there or not, but they tell you the probability, the predict let's say the risk that if this is high or low. Then you have a predictive one, so where let's say you can have some sort of a treatment is ongoing, and then you want to see that if the person is responding to that treatment or not. Okay, sometimes it is not visible by the symptoms that the person is having, and so then you can have predictive biomarkers which help you to determine that if the treatment is getting effective or not, and then these help us to see if the same treatment should be continued or something needs to change. Okay. And then you have a pharmacodynamic one, so which is basically for your drug response. Okay, and yeah. Now, in terms of types, so as I mentioned, so you can have DNA or RNA or protein or metabolites. So in terms of DNA, it can be in the form of mutations, SNPs, or methylation status. So for example, in glioblastoma, so you have MGMT promoter methylation, so which is a classic biomarker for methylation in for detecting glioblastomas, right? So in all glioblastoma, more or less, you have this promoter of this particular gene is methylated, right? So then you have RNA, so where you can have like gene expression levels, splice forms, or non-coding RNAs. And in proteins, you can have like enzymes and receptors. So for example, in prostate cancer, you have PSA, yeah, so which is like high in prostate cancers. And then you can have metabolites as well. So where like small molecules, they can reflect your metabolic state. Okay. Now we are talking about RNA-seq, right? So like RNA-seq, like why we are covering this, because like RNA-seq is also like kind of, you quantify the expression levels, right? So RNA-seq is also, in a way, it can be a biomarker, right? Where the RNA changes, they quickly determine the cell fate and they reflect the disease dynamic better than the static DNA. Yeah? So DNA is static, right? Whereas RNA, it changes, from like depending upon a state or if a person is diseased or not, right? And it can capture both coding, non-coding RNA, even the fusion events and splice events, right? Now, an example for you. So if, let's say, imagine a patient is infected in influenza, so what kind of biomarker would you use to best capture this dynamic immune response? So based on all of this information here, right? Which one do you reckon, like, would best capture the immune response in a patient with virus? Anyone? Okay, why DNA? RNA, yeah, why either of them? Yep. So in this particular case, it would be RNA. Yeah. Yes, but what we try to capture is the immune response. Yeah. So yes, so we could do DNA as well, but in this case, we want to capture the immune response, so which we would do based on, let's say, the viral load. Yeah. So for example, like, so RNA would actually give us the. So you see here, it's already there in the dynamic state. Yeah. So RNA actually better reflects the disease dynamic state, and that is what we are trying to capture over here, right? The dynamic immune response, which you would not really be able to capture efficiently in DNA. Okay. And also, like, I think there is one biomarker which is also, you guys have also probably gone through like 3-4 years ago. So what do you think? So in the COVID tests, right? So you all like would have heard that time about RT-PCR and stuff, right? So yeah, I think you guys were not in, you guys were still in high school, correct? So which biomarker was used in this RT-PCR tests for COVID? Anyone? For COVID tests, which kind of biomarkers were used? Which type of these 4? Yeah. Yep, exactly. RNA. Yeah. So it's all the clues also in the RT aspect of it, where you have the reverse transcriptase. And what do you do reverse transcriptase for? RNA, right? To convert RNA to cDNA. Okay, great. So yeah, so now going on to the main focus of this internship, that is RNA-seq. So what is RNA-seq? So as like by definition, it is a high throughput sequencing method to profile the entire transcriptome. So like in genomics, you capture the entire genome, right? So in RNA-seq, you capture the entire transcriptome, right? For a given species at a given particular time point, right? Because RNA is something unlike DNA, which can change from tissue to tissue, time to time, and also under different environmental or different conditions, right? So whenever you do RNA-seq, so these are the things like you have to keep in mind that you can capture the entire transcriptome at that particular given snapshot of time or condition or organ. Okay. And it can measure both like quantitative, basically like the expression levels, and also qualitative, which can actually mean like the different isoforms or fusion events or the novel RNAs as well. Okay. And so in terms of biomarker discovery, so as a very like crude example, so what you could do is you can compare expression profiles. So you can do RNA-seq for a healthy sample and a disease sample, and then you can identify the genes which are highly expressed in the disease samples, right? And then you can do some sort of a validation of these genes, like especially take using a larger cohort. And so once you do that, so then you know that genes which are highly expressed in the disease that you are interested in. Okay. And now once you have identified these and also validated, you can actually use these in in a diagnostic setting. So where just like based on the expression levels of these gene, you can say that okay, a person is diseased or not, right? So if that is not already evident from your symptoms. So here, for example, in the case of breast cancer, so there is one very popular gene which is called as HER2, which is highly overexpressed in breast cancer samples. So let's say, so in this particular case, so HER2 gene, it is a predictive biomarker for your for breast cancer, and it is also a therapeutic drug for like this particular therapy. So in breast cancer also, like there are multiple types. Yeah, so there is HER2, then there is BRCA mutations, and there are some fusion genes as well. So there are different types of breast cancer. Yeah, so the type is determined by what is the underlying cause. So in one case, there is this overexpression of HER2, and then there are also mutation in the BRCA gene. Yeah. So, and then for depending on what kind of breast cancer you have, so there are different therapies which are available at the moment. So there is one therapy called like this one, trastuzumab, which is specifically designed for those breast cancer types where the HER2 is overexpressed. Yeah. So if a patient has a breast cancer and they have high, and that is because of HER2, so then they are put on this particular therapy. Okay, and this is how in real life, like we try to let's say use these RNA biomarkers. Okay, and yeah, now okay, this is my second last slide. So last question for this talk for you guys. So if you have RNA-seq and you identify like more than 2,000 genes which are overexpressed in your disease, then how would you prioritize like which ones you should validate as biomarker? Any ideas? I'll give it a think. Anyone? Anyone? Feel free to unmute and speak. Any guesses? Okay, fine. If not, like, yeah, I think I'll move and maybe I'll take the questions after this. So first of all, like, it should be consistent across the different cohorts. Yeah. So sometimes, let's say you have samples from, let's say, Indian population or like some other populations. So in all of these different cohorts, like, it should be consistent across them. And also it should be unique to your disease. So it should not be that, let's say if this particular gene is high in one, but it's also high in other diseases also, then how do you know, like, which disease it is responsible actually for? Yeah, it might be just upregulated in all diseases. So you have to be do that, like it has to be unique to your disease or condition that you're studying, right? And there should be some valid biological significance around it as well, right? And it should be also like bit clinically feasible. So this is mostly due to like the sequence of the genes. So mostly it is fine. Yeah. So you should be able to design antibodies around like certain proteins as well. So but in terms of RNA, it's like not a big problem. Yeah. So these are like some of the things you have to consider if you have to translate a particular RNA-seq biomarker or any biomarker for that matter into clinic. Okay. Now, yeah, so this is the last slide. So as a take-home message from this, so what did we learn today in this session? So we see that sequencing has evolved over years from Sanger to NGS and to long-read platforms, revolutionizing genomics. And then the biomarkers as measurable indicators for disease state, outcome, and therapy response. And then, this RNA-seq also provides an unbiased. So remember, like this is when we do RNA-seq, so we do not like select any transcriptome. So we profile the entire transcriptome. So we do that in an unbiased fashion. And yeah, so this can be used for, detecting RNA biomarkers and some real life examples from HER2. And also, for example, in the case of even like your COVID tests as well. And in the next session, we'll talk about we'll study some RNA-seq experimental design and analysis workflow. Okay, with that, I will stop my screen share and take few questions. And yeah, feel free to unmute and or write in the chat box your questions. Or everything was clear? Yeah, was it?
Dear participants, is there any questions to be answered? Is there anyone trying to unmute? No. Okay. Yeah, a quick thumbs up says the session is clear.
Yeah. Or feel free to use the Google Classroom as well. So you might not have the questions now. Okay. If there are more questions, feel free to use the classroom as well. We'll be happy to
Small correction.
address them over there. Yeah.
Technical questions will be posted in Telegram discussion forum.
Okay. Can you add me? Can you add it to the Telegram? Wait.
I
might have. I might have an uninstalled Telegram, or it's been a while I opened it up. Yeah. Can you just add me to the Telegram forum as well?
Sure. Yes, no questions.
No.
Okay. Saurabh, do you want to say any prerequisite before next session? I think next session will
Not
be
for the next one. No, not for the next one. Next one we don't have anything.
It will be starting in an hour, right? It's 11:45 Indian time.
Yeah. So we'll have it at
1 p.m.
Yeah, 1 p.m. Yeah, I mean, we can start at like 1 p.m. or even like 1:45 if you guys want. But up to the participants.
It's again a 2-hour session, right?
So yeah.
Would you like to take it 1? I think that's what we meant.
Yeah, it's fine. Fine. Then let's do it 1.
Let's go with the schedule.
Sure. Okay. Okay. If no other questions, then maybe I'll leave you guys a bit early, so that give it a think. Okay. Like if you have any other question, feel free to write in the classroom and stuff as well. Maybe I can take them up in the first 5 or 10 minutes of the next session as well.
Sure. Thank you so much.
Okay. All right.
Thank you for making this inauguration session a successful one and delivering a better RNA-seq program for all the participants. Thank you, Saurabh. And dear participants, thank you all for joining us today. We have shared the feedback link in the chat. Feel free to post your concerns. If you need us to focus on anything like phase of the session, or if you want us to deliver it in a different way, feel free to post the same as well. You will be receiving the session recording shortly. And Saurabh, may I know when you can post this material in Drive?
Sure.
You will be posting?
Yeah, you want me to do it now or after the second session?
After the second session, editing
okay,
feasible.
Sure. Yeah, sure. After the second session, then I'll do both the slides.
Yeah, fine. Okay. Perfect. Thank you.
Yeah, and Ms. Bhavani, I have one request. Kindly inform that feedback will be considered as attendance, okay? Yeah.
Yes.
So every day we take feedback, and whoever give the feedback will consider as attendance.
Thank you, Mahesh.
Yeah.
Dear participants, as mentioned, I'd like to say feedback will be considered as your attendance form. So please do fill it out without fail. With this, let's wind up the first session. We will be meeting around 1 p.m. Indian time and requesting you all to join the session on time. Thank you all, and thanks once again, Dr. Saurabh.